Good afternoon. This afternoon, yeah, it's kind of a reschedule, and today we're going to talk about metabolomics and proteomics, which actually was planned, I guess, on tomorrow. Anyway, I didn't know, so anyway, it's been a replace here. I'm going to talk a bit about the metabolomics data, and in specifics, how to pre-process the metabolomics data, because it's not easy to get hold of it. Well, it's easier to get hold of it than to process it. And I want to discuss a couple of issues with you that you can come across if you do it. So if you handle metabolomics data. So I'm kind of interested in who of you have already experience with metabolomics data. Okay, and who of you are really bioinformaticians? So. Okay, and who have a biological background, like in that's most of you. Okay, okay. So that's just for me, because I won't be talking about the data analysis of the metabolomics, because the next lecture will be for me will be component analysis and combining data sets, so data fusion, so which is more of a data analysis issue. But I won't go into the details of the data analysis, just the pre-processing of metabolomics data itself, which is hard enough. So what will I be talking about? Well, first of all, a small introduction about the very common separation technique, HPLC, and mass spectrometry, which is our detector. The pre-processing steps, some of them, I won't go into detail of all of them. So what can go wrong and how to fix it, and then a hands-on, like I made a small metabolomics data set, and really a couple of metabolites, and you can try to answer the questions that I created with them. But my presentation will be followed directly by Andreas about proteomics, and actually it's also kind of a logical flow, because I will be talking a bit about mass spectrometry and the whole separation technique in front of what is necessary to identify and to identify them, and of course that will be used in proteomics as well. So what does metabolomics entail? Well, first of all, it's a scientific study of chemical process involving metabolites, which sounds fair enough. So what does the metabolome represent then? Well, it's a whole collection of all the metabolites in a biological cell, tissue, organ, or organism, which are the end products of cellular processes. And what are the specifics? First of all, they are small molecules, about 1500 Dalton in size. So what do they contain? Well, lipids, sugars, amino acids. What could they be? Hormones, other signaling molecules, and secondary metabolites, for example, in plants. Metabolomics, not related to growth, development, and reproduction. So if I look at the omics groups in the biological context, so bioinformatics and systems biology, then every omics group has its own way of detection. Genomics, for example, well, you've learned about that yesterday. Well, it's about sequencing methods. Transcriptomics, also sequencing, but also microarrays. Proteomics, it's mass spectrometry and NMR. And metabolomics is predominantly mass spectrometry. And that's what I'm going to focus on. So, what are the main separation techniques? Actually, in metabolomics, we've got gas chromatography or ultra-performance liquid chromatography, and the detection, which is NMR or mass spectrometry. Well, the ones that I underlined are the most occurring ones in metabolomics. So what does liquid chromatography entail then in this case? All of you have done some analysis, really like sample preparation, and anyone familiar with it? Because there's a lot, only a few of them. There's a lot what can go wrong with it. And I'm always, I always, well, am surprised by what these analysts can come up with, because I've done it a couple of times myself, and I'm not able to reproduce these results. So it's really hard to do it. So what is it? Well, it's an analytical chromatographic technique for separating ions or molecules that are dissolved in a solvent. So the sample solution is in contact with the second solid, a liquid phase, and in our case it's always a liquid phase. And the different solutes will interact with the other phase to differing degrees, and due to these difference in adsorption, ion exchange, partitioning, or size, and that interaction allows the mixture components to be separated, which is exactly what we need it for. So in a schematic, which we got from this website from Waters, which is a renowned mass spectrometry and HPLC producer, here we've got the solvent in which there is the actual sample itself. We've got a pump. Well, it's a solvent, and here's a pump, and the sample is injected into a continuous flow, which is then pushed through this HPLC column, and then arrives at the detector where it is detected, and it actually is detected, and it generates a signal. And this signal versus time is what we call a chromatogram. And actually this is very, very important, because this is our base of our data analysis and of everything, this chromatogram. Because actually this peak is some kind of in relation to the number of the amount of compound that I'm looking at. And in the end, what is not needed will arrive in the waste. So if I look at it schematically, so in the column itself, so what happens? So if we look at some kind of sample which is injected, and I've represented it here with a black band, and in time, while time progresses, there is a continuous flow of solvent. Actually what we will get, because of this ion exchange and adsorption effects and everything, I get a separation between the compounds that are actually in my sample. So that's actually what we want, because then I've got a nice separation. Then in time I can identify or quantify my compounds of interest. So what is a chromatogram then? It's a representation of the separation that has chemically occurred in the HPLC system. Actually, this presentation is online, but no, I didn't put it in there, so you can always look at it later if you want to. And a series, well, in time, what you will see is a series of peaks rising from the baseline. So every compound will generate a small peak, and that will be my chromatogram. And each peak represents a detector response for a different compound. So, and this is really different. This is really important. A detector response. So for a different compound. And in the end, in time, I get a nice peak like this. So if I look back into my whole system again, I've got a couple of detector types, and mass spectrometry is the one most used in metabolomics, and that's the one I will be focusing on, or we will be using. So the summary here again, everything I put in here, it reaches the detector, I get my chromatogram, but also I get some kind of well, you see here, every peak is a different compound, so they were identified. Well, how were they identified? That's the main thing, and that's where we come to mass spectrometry. So that's an analytical technique for the determination of the composition of a sample or molecule. It's also used for elucidating the chemical structures of molecules such as peptides and other compounds. And more, I guess, about elucidation is in your talk, because I won't go too much into there. And actually the whole principle is based, or it consists of ionizing chemical compounds to generate charged molecules or molecule fragments. And what we will detect is their mass to charge ratios. So if I look back on my sample, so the one I got from my HPLC, so it's separated, it will reach an ion source, I get a mass analyzer, so it will reach the detector, and I get a mass spectrometer, mass chromatogram. So I've got here several masses, and every peak here is specific for a specific compound. So we've got two dimensions already. We've got a time dimension for separation for the HPLC, and we've got another dimension which separates the compounds and with at every time point. So I got a two-dimensional data set. So then if we look at the hyphenated system, so the combination of LC with mass spectrometry in this case, here we got my HPLC or LC, my mass spectrometer, and actually in conjunction or hyphenated, combined, we get this whole flow. And so if again, we will look at it schematically, I've got my sample, it will go through the column, which will separate them. I get a fraction at time point X. Time point X, it will reach a detector. I get a whole mass chromatogram. And this one is measured for, let's say, 5 minutes. And that one actually, and this is really an issue, or this we need to you need to understand if we talk about mass spectrometry. This one is what we call the TIC, the total ion current, and actually that's the sum of all intensities of all masses that I've measured at this time point. And so that's a lot of masses and a lot of compounds that come from the column at the same time. So if I look at my tick again, here's a tick in the time. I look at two subsequent time points at 120 seconds and 121. I got almost a similar mass spectra. So this is about the data set that I'm going to use, or the way the data is generated. Now actually it's all about the data processing. So now what can I do? How can I generate this into some data that I can really use for data analysis? And actually, if I look at my whole metabolomics workflow, it actually consists of a whole lot of steps. So first of all, I need to define my biological question, the experimental design, the sampling and the sample preparation that have to accompany this, the data acquisition itself, which is very elaborate, data pre-processing, then the data analysis, and after that only the data biological interpretation. Well, this one, sometimes if I do the data analysis, then I figure out, oh, something has gone wrong in the data pre-processing. And if I look further in the data pre-processing, I can figure out that it was something in acquisition. And sometimes even there I can see, hey, something went wrong there. So it's also a cascade back. So it's a whole complex system. And it's not like I go from one side to the other. It can also be from this one step back, then continue again. And but data pre-processing is a very important step in this whole pipeline. So how do we get from the raw data to my peak table that I can use for my data analysis? That's what whole pre-processing is about. And in here we see this peak label, so it has at 4 minutes, for example, retention time, it had this area, and it was this metabolite glucose. So now how do I correct my data if I have my data? Finally, if I have my raw data, so how do I get my data? And there is some need for corrections, and I mentioned a couple of them here. First of all, there's preparation and accuracies, like weighing or pipetting, so some really analytical errors. Also, we call them gross errors, but because these analysts have to measure loads of samples, it's almost hard to call them gross errors, but they can sometimes forget to add internal standards or add some compounds, or in wrong volumes, or labeling errors, which often happens. Then we have some machinery errors, like chromatographic errors, like a carryover and background effects. Like, for example, I've got a sample which had contained a lot of, for example, compound A. Then it can happen that in the next sample it still has some residue from the previous sample. so I will have a larger peak because of it. So these are all things that you have to take into consideration when you use this, or when you do this kind of analysis, or when you do pre-processing, but also data analysis. Then we have got things like column aging. So a column wears off in time. Actually, the response, so the separation becomes less. So that's a big issue. Column changes. So after it's been aged too much, we can change it for a whole new column, which again I can get completely different results. So you have to be aware of that. Then solvent changes. It will affect it. It can affect your results. Then we have some detector limitations, like how much is in my sample, how much of the compound is in my sample. Is it just or below level of quantification? Or if it's too high, then perhaps it will saturate my detector, so I have no usable signal. Then I've got some integration errors, like if I have this peak, sometimes because it's an automated process, if I have loads of peaks, then I will have selected the wrong peak. Can I select the wrong peak? Or my peak is not within my parameters that I've selected to integrate. Or I didn't use a peak integration method. So I actually, well, it's not visible here, but there's 3 levels of correction. And actually there's also, well, the first basic level, which is really the raw data level. I'm not going into that right now, because this is really mass spectrometer specific. So, and so then I will talk about sample level correction level, like integration, the things that we can see and that you can visualize, calibration lines, and the use of internal standards. So if I have this chromatogram, I can try to integrate it. And integrated is, if we look at this peak, it would seem trivial enough, because this is one peak, and I can, if I integrate the area below it, it would give me my let's say my relative concentration, or some kind of an indicative amount for what's in this for the compound in this sample. But if I look closely, I can already see that I have 2 peaks here, which are not separated here. This is already very difficult to automate this, because sometimes in the one sample this will look like 1 peak, sometimes it's 2 peaks, they're separated, sometimes separated. So this is already really difficult, and it looks really trivial. But the results can be very problematic if you sample to sample. And in here I've shown some examples, like what can go wrong. Well, sometimes it takes the center one, sometimes the left one, sometimes the right one. Sometimes it will take this one, but you pointed this one, but it will take a whole different one. Sometimes it takes the left one, sometimes the right one. So there's no way in telling what will happen. So this is automated software. So this is a real issue. So if you do wanna do the data analysis and the pre-processing, these are the things you have to face with. So that's a real problematic, at least in the metabolomics pre-processing pipeline. So is it a big problem? Well, if I know what I'm looking for, I can resolve it. I mean, I can spend some time to do it, but it takes a lot of time to fix it. So 1 to 2 days per batch, which is really costly. But I get software updates from vendors, and it will work. I mean, in the end, everything will be sorted, and this will take a couple of years, and then everything is fine. So, but it's time, but it will work. But if I do untargeted analysis, so I'm not sure what I'm looking for, which is actually the most interesting in the research field, because we want to do some data discovery, some discovery of new compounds. So we don't know what to look for, then it's a big issue. At least if we were also, because we got loads of compounds and we want to do some data analysis, I would really like to know whether or not I'm looking at the same compound from sample to sample. So that's an issue. So but at least, well, there's lots of software available, and it's still under development. So, and also, well, we have peak areas. So if you think that this is only the thing, now we have the peak areas, but like I said, there's also retention time. So at specific retention time, we have a specific mass which elutes from the column, and from sample to sample, we have small variations, and they can be really stupid, like a temperature in the laboratory. And actually, we've seen that, so we could see the temperature. We could see the temperature rising from the peaks that we saw, so the peak areas and things like that, and the peak shifts. Also, the atmospheric pressure and the pH, the time and composition of the samples in themselves. So there's the column degradation. We got it again. So it's hard to control these samples, or to control these measurements. Here, for example, we've got retention time shifts. So like here, we got the, well, like here, but the thing is here, in this case, I've got some kind of an equidistant separation. So our separation, actually time shift. What we also find is that the shift here is much larger than there. So we have to correct for that as well. So it's a non-linear time shift, which is kind of an issue. There's of course some algorithms here, and we've got some automated tools which are called correlation optimized warping, or dynamic time warping, or parametric time warping. So there's lots of things to align them to each other. So, well, the global working procedure is to split the target spectrum up in multiple parts and shrink and stretch parts in such a way that similarity with the reference spectrum is optimized. So we have to select one spectrum as a reference, one sample as a reference. So if you think we're already there, then, so what are the caveats with when using mass spectrometry actually? So we need some internal standards, and why is that? Because each metabolite has its own response factor. So the signal depends on the number of molecules, but also on the type of molecule. So if I have a signal of 1,000 ion counts for one compound and 1,000 ion counts for another compound, you would think they have the same concentration, but it's not. Because it also depends on the factors like solubility, ionizability, fragmentation, mass discrimination at the detector level. So and furthermore, it's also matrix dependent. So also it depends on what else is in your sample. So, and because we have and which is even further complicated if I use my LCMS, because my matrix is not constant over time because of the separation. So I also introduce a lot of variation and complications this way. So I think it's always interesting to find in the end that we can still measure with it and adequately, and that we can still have some nice results with it. And internal standards is one of the issues, or one of the items with that we can use for correction. So if we look, for example, at this one sample, and I have here 4 peaks, A, B, C, D. So we have different areas under the curve, but it doesn't mean that my concentration of D could be, or it does, yeah, it could be that concentration D would be larger than A, but C again would be larger than D. So that's really non-indicative. Also, if I have different matrices, for example, from sample to sample, even if we're talking about the same compounds and they elute at the same retention time, then still it doesn't mean that these concentrations have to be the same. So that's really an important issue. So what we do therefore, what we therefore include is an internal standard. And why? Because we can monitor and correct variation for the, well, that have been introduced during sampling and sample preparation. But also other variations like pipetting, centrifugation, derivatization, and how does it do it then? Well, because the chemical behavior should be as close as the analyte that you're interested in. So in mass spectrometry, and it provides its own ions. So, and usually it's a stable isotope, a C13 or an N15 compound of the one that I'm interested in. it could be also isomers, homologs, or structural analogues if you cannot find the appropriate stable isotope. And the idea is that the sources of variation should affect both the sample and the internal standard equally. So, and here I actually plotted here one. So what I did is for every compound I added an internal standard specific to that compound, and in the end I will end up I can calculate the ratio between them, and that should be constant between the samples. Yeah. Yeah, but that's an issue. Yeah, that's a real big issue, because there's no way in telling which compounds you will find, and I think that's a very important point. So usually what you do is you take a set of set of compounds that you expect to be in your method, what you expect to detect, and then you, well, okay, for this, like in lipids, for example, you say, well, this is a triglyceride, so you have this one, and for the diglycerides you have this group, you have this reference, and that's the way you do it. You should, of course, make you shouldn't make it too complex as well, because if you add a lot of internal standards, you influence your own measurement again, and you will easily get detector saturation again and things like that. So that's a hard thing. So we are busy. I had some nice experiments, and they were working on it right now, and they show some nice results to do it in a different way. And but well, I can talk to you about it later. So yeah. Okay. Yeah. Yeah. So what was the idea? Well, that the ratio is my relative concentration, and actually and it's my peak area unlabeled, so my compound divided by my internal standard. And what is the idea? Like here I have 3 calibration lines, one in water, that's the top one, the other one in water plus some sample preparation, and the third one, it's a lower one, it's in urine sample. It's in urine actually. So, and what if here, this is the original calibration line, but now corrected for my internal standard, I can see, well, they are closely. They actually follow the curve perfectly. And that's the whole idea, to make them comparable. So, and remove the variation introduced by sample preparation, analytical variance. So another issue then, or how we can get some kind of measure of error, is to look at repeated measurements. So, and actually what we will find, at least for mass spectrometry data, is that if I increase my analyte concentration, that the error will increase also proportionally. So actually it's larger here, my variation is much larger here than it is here. So actually what you can do is we can create some kind of we can correct for it using some kind of weighted regression, where the weight itself is an inverse of my variance based upon this triplicate measurements. Actually, that's why you should always measure calibration line in triplicate. And in here we see, for example, a perfect calibration line with this regression coefficient. But if we want to correct for this heteroscedasticity, then we can use this weight factor in there, and we have a weighted regression line, and where we can still see that we have a nice calibration line, even though my variance is increasing here in the larger concentration points. Then another thing to consider is the linear range of my detector itself. So a saturation of detector, so after this point you're not allowed to measure anymore. Well, you're allowed to measure, but you're not allowed to include your results in the analysis anymore, because this we cannot correct for it anymore, and it's not reliable results. So, and this is the effect. Actually, you see that below this area we fall out of my scope, and above this area here we see that we don't fall in the linear range anymore. So then finally we are arrived at batch level, because now we think, well, we are already there, well, we're not there. So we also have some corrections at batch level, and what do they correct for? Because an internal standard cannot correct for dilution effects of the biosample. For example, urine. I can have effects from urine to urine sample, and urine is a real notorious one at that. If we have for many compounds, we only have one internal standard per class of compounds, like you mentioned. Yeah, so there's not really always a perfect internal standard. So also you should note that using an internal standard can really ruin your results. So if you don't have the right internal standard, then it's sometimes advised not to use it at all, but in rare cases. So it cannot correct for things like column degradation, different machinery, and things like that. And therefore we need another type of normalization, and actually we use some kind of reference samples in this case, and we call them quality control samples, which is usually a mixture of all the samples in my study that I wanted to analyze. So usually I set aside part of it, and then I make one mixture of it, and so I've got a bulk of it, and then I can make more samples of it, which I can measure repeatedly, and of which I know should have the same response over time, and also on different machineries. So, and the hypothesis here is that the reference sample, they should behave the same over time, and the ratio over my internal standard should remain constant. So, and also we can also do that with replicate injections of samples as well. If you include replicates in a smart way, then I can also identify some nice I can also identify some nice analytical errors and things like that, and correct for it. So if I look for example, in this case, I've made a plot of the response. In this case, this is for one metabolite, and these are real samples, the blue ones, and this is the measurement order, and this is a response. So these are different samples, and actually I included my quality control samples, and I see actually from batch to batch, and this one, and a batch usually consists of 150 samples. And because that's how much can be prepared in one go, or things like that. Sometimes it's less, sometimes it's much less, sometimes it's more, but 150 already is a lot. But what you see is let's assume this is one day, you see that during the day this response of one variable of one compound, while it should remain the same, it increases. So it's fair to assume that would also be true for your real samples. So they also have this, they also suffer from the same influence, same trend. So, and these were replicate study samples. So actually what was devised is some kind of, usually these quality control samples, they were used to monitor this behavior. And actually we could also use them to correct for variation, and actually what we did is we drew a line through it, we actually penalized smoother in this case, and we actually followed this pattern, and actually what we said is, well, okay, they should behave the same, so let's make them behave the same, and then project it on there and remove the analytical variation because of it. So, and now every metabolite here, for every sample here for this metabolite has been adjusted accordingly, and I have actually corrected for my analytical variation over time, or degradation effects, or things that I could not specify earlier. So this is for before, this is after. So, and one way to check actually is the quality control, the RSD values of my quality control. So this is before and this is after. So you can see that the smaller variance between the samples. But of course, this is what expected. Actually, but we also can do that with replicates, and I would also see an increase here. And I see in this case the increase is a bit smaller than expected, but it is getting better. So, and this is real. This is a real nice external validation of my quality control check. In this case, my quality control correction. So, in summary, for pre-processing mass spectrometry data for metabolomics in this case, well, we got the integration, some semi-automatic integration. We've got some corrections. We got a blank correction, internal standard correction, intra and inter-batch correction. And then we have a quality control report we can create based on what I've did here. We can detect, inspect, and correct deviating samples. And when necessary, we can repeat that again. Then remove metabolites measured with insufficient quality. So that's also for untargeted analysis from the data and export. And when possible and required, we can regress and quantify. So if I have added my my appropriate quantify calibration lines. So finally, I can fill my table with my numbers. And I want to acknowledge all these people here which have been working very hard as well on this data.